like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Hi, welcome to the National University of Singapore Yong Lu Lin School of Medicine's Healthy Longevity webinar. I knew I was going to forget to mute myself at one point in one of these shows, and I almost did it there, but I think we made it. Thanks for joining us. We have Professor Seung-Jae Lee from Korea, who will be telling us all about aging in a few minutes. I'd like to remind you to use the Q&A function to send questions, and please send questions tonight because we have Charmaine back assembling the questions. She's a graduate student in my lab, and if she has to make up her own questions, they're going to be extremely hard, and Sanjay won't be able to answer them. So we need questions from the audience tonight. But before we get to Sanjay, we're going to hear from Sarah May Kapinpin, research assistant in the Center for Healthy Longevity, and she'll be telling us a lesson from a non-aging species you may or may not have heard of, Hydra.
Thanks, Prof. Brian. As mentioned, I'm Shara, and I'll be sharing with you a little lesson we can take away from these little guys called Hydra. Hydra was named after a mythical creature in the Greek legends, often described as an immortal, gigantic, water snake-like monster with 9 heads. In the stories, if anyone were to behead the creature, they would find 2 more heads would emerge from it. This Hydra may not be as fearsome as it is in the stories, but it is definitely immortal, or what we call negligibly senescent, and can in fact grow a new head if cut off. But before I go on to talk more about Hydra, let's look at some of our classical animal models that have helped us understand the biology of aging. Most of our current knowledge on mechanisms of aging, aside from human samples or cell lines, have been acquired using classical model systems such as mouse, Drosophila, and C elegans. There are many advantages to using classical animal models, such as they are easy to handle, have short generation time, and have well-established husbandry protocols. We also already have a wealth of information on these animals at our disposal. So you might say, with all these advantages, is there really any value at all to look at non-classical animal models like Hydra? One clear advantage is that by looking at long-lived animal models, we might be able to learn a thing or two on how they maintain their long lifespan. So Hydra are freshwater polyps from the Cnidaria phylum, the same family as other long-lived negligibly senescent animals like jellyfish and corals. Hydra can reproduce by asexual budding, but can also be triggered by specific environmental conditions to produce gametes for sexual reproduction. In Hydra, there are 3 main stem cell populations, namely the endodermal epithelial stem cells, which gives rise to the inner layer, ectodermal epithelial stem cells, which give rise to the outer layer, and finally the interstitial stem cells, which is the subject of interest for many research in Hydra. So both the endodermal and ectodermal epithelial stem cells are able to generate differentiated somatic cells every 3 to 4 days. Interstitial stem cells, on the other hand, are multipotent stem cells and can generate not only somatic cells, but also germ cells such as your sperms and eggs. These cells are able to generate new ones every 24 hours. So many studies in Hydra have demonstrated responses to the known hallmarks of aging. But for today, we will only look at stem cell exhaustion and telomere attrition. So in a study conducted by Galliot and her team, they found that in Hydra, FOXO, a well-known longevity gene, is upregulated in the interstitial stem cells, more so than in other stem cell populations, and that the ablation of these interstitial stem cells sees a stark reduction of FOXO expression in other body column cells. They also found that interstitial stem cells upregulates genes that maintain telomeres as well as DNA repair mechanisms genes that can ensure the genome integrity in these cells. These genes, such as TERT and TERF-like, can be seen to be more expressed in interstitial stem cells more than other stem cell populations. This alludes to the Hydra's ability to maintain self-renewal in their stem cells. Adult stem cells also face the challenge of maintaining tissue homeostasis by self-renewal while maintaining their proliferation potential over the lifetime of an organism. Continuous proliferation can lead to telomere shortening, which compromises the genomic integrity of stem cells. So to prevent stem cell exhaustion, there is a need to maintain a pool of quiescent stem cells that divide only when necessary. In the study done by Ganikar and his team, they pulsed Hydra with thymidine analogs and monitored the presence of these analogs over the next few days and weeks. They found that Hydra reported heterogeneity in the cell cycle frequency of their stem cells, and this demonstrates that there is a subpopulation of stem cells that divides infrequently. Slow cycling stem cells from all three lineages can remain undivided for 8 to 10 cell division cycles and can still re-enter the cell cycle thereafter. They also found that these slow cycling interstitial stem cells are in close contact with extracellular matrix, demonstrating the importance of these ECMs in maintaining stem cell niche. These slow cycling stem cells, like in mammalian stem cells, can be activated to undergo cell division during head regeneration within an hour after amputation. So these findings are just one of the few that demonstrates Hydra's anti-aging mechanisms. There are many other studies that points to Hydra's ability to tackle some of the hallmarks of aging. But what are the advantages of using Hydra, you might say? Other than being simple and easy to maintain, Hydra can also provide us a unique opportunity to study mechanisms in animals with extended lifespan. Of interest are these two species, Hydra vulgaris and Hydra oligectis. Hydra vulgaris have been known to demonstrate little to no senescence, exhibiting stable budding and gametogenesis properties over several years. Hydra oligectis, on the other hand, showed a rapid and dramatic reduction in their budding rate and appeared exhausted only when undergoing sexual differentiation. So by comparing these two species, we may be able to delineate the mechanisms of aging in a long-lived versus short-lived organisms. Hydra's amazing regenerative capacity also means that they can be subjected to a plethora of experimental manipulations that are not possible in other models. Moreover, working on novel animal models like Hydra, there is a potential to find novel compounds that can be used to emulate the slow aging ways of Hydra. And that's what we've learned so far from our non-aging friends Hydra. However, we do not yet know if any of these could be applicable to higher complexity mammals like us. There is definitely a lot of work left to determine its translatability. Thank you.
Thanks, Sarah. So I want to get on to the main feature tonight. And so I want to introduce Seung-Jae Lee, who's a professor at KAIST, the Korea Advanced Institute of Science and Technology. I've been there a few times. It's a fascinating research institution. And so there's a lot of great research going on there. He's been working on aging and longevity using C Elegans and mostly as a model system. And in particular, he's focused on the role of insulin IGF signaling, mitochondria, carbohydrate and lipid metabolism, and RNA quality control in aging and healthy longevity. Tonight, he's going to be telling us about molecular genetic approaches for healthy longevity using the worm Caenorhabditis elegans. Thanks for joining us, Sanjay.
Thank you for the introduction, Brian. I'd like to start my talk by thanking Brian for inviting me to present our recent work here. Today, I'll be talking about two of our recent papers about molecular genetic approaches for healthy longevity using Caenorhabditis elegans. My research group has been interested in aging, and I think many of the audience members are too. Perhaps the most important goal of aging research is to promote long and healthy human lives. Because humans are mammals, to achieve this goal, it is very important to study aging in mammals. Although mammalian systems are crucial for aging research, one limitation for studying aging with mammals is that aging research takes very long. So we need a system for studying aging in a relatively short time as well. For that purpose, C Elegans is very small nematode worm, 1 millimeter long, is one of the best model systems for aging research. The biggest advantage of C Elegans as a model for aging research is that it has a very short lifespan, 3 weeks, and we can observe aging processes within just 2 weeks. In addition, we can manipulate genes very easily using a variety of molecular genetic techniques such as mutagenesis screen, transgenesis, and CRISPR-Cas9 genome editing. Importantly, many genes that influence aging and lifespan in C. elegans are conserved across the phyla. Using this C. elegans as a model system, my postdoctoral advisor, Cynthia Cannon, who is now a vice president of Google founded company Calico, showed that a point mutation in a gene called DAF2 can double the lifespan of C. elegans. You can see that, as I told you in the previous slide, wild-type animals live around 20 days in average. DAF2 mutants live over 40 days in average. This is very important finding because from this finding, scientists were able to think about doubling animal lifespan and delaying aging, perhaps including humans. What is DAF2? DAF2, it turns out that DAF2 encodes a homologue of insulin and IGF-1, insulin-like growth factor 1 receptor, suggesting that hormones such as insulin or IGF can regulate aging. From these and other findings, now it's obvious that aging and lifespan can be genetically regulated. Subsequently, many genetic and environmental factors that regulate aging have been identified, including insulin IGF-1, targeted rapamycin, dietary restriction. As I said in the previous slide, many of these factors have been shown to be evolutionarily conserved. This review paper published by David James and Linda Partridge shows two of the most evolutionarily conserved aging regulating pathways, insulin IGF-1 and targeted rapamycin signaling pathways. Like C. elegans DAF2 mutants, heterozygous knockout of IGF-1 receptor or fast-specific knockout of insulin receptor delays aging and extends lifespan in mice. In humans, polymorphism in two genes, IGF-1 and FOXO3A, a key transcription factor acting downstream of insulin IGF-1 receptor, is associated with longevity. So genetic variations in insulin IGF-1 signaling pathway genes likely underlie human longevity too. In 2011, Walter Longo's group published a paper, and they show that Laron syndrome people who have mutations in growth hormone receptor are short, but they are almost completely free of two age-related diseases, cancer and diabetes. And this was shocking, and it was featured on the science section in New York Times. And this is very relevant to this pathway because in mammals, growth hormone acts upstream of IGF-1. All these findings originate from C. elegans DAF2 mutant study, and therefore we believe that studying C. elegans aging can lead to insights into human aging and human longevity and age-related diseases. These are representative factors that regulate C. elegans aging. Among them, we have been mainly working on the role of mitochondria, nutrient, temperature, sensory neurons, and insulin IGF-1 signaling in aging and longevity. We have recently started working on the role of novel non-coding RNAs and mRNA quality control in longevity. But today, I'm going to present two of our recent papers regarding the role of insulin IGF-1 signaling in the rejuvenation of immunity and healthy longevity in C. elegans. This is a simplified insulin IGF-1 signaling pathway in C. elegans. Briefly, mutations in DAF2, insulin IGF-1 receptor, leads to the inactivation of AGE1 PI3 kinase cascade, and this in turn causes the activation of several anti-aging transcription factors, including DAF16, FOXO, SKIN1 NRF, and HSF1 heat shock factor 1. The effect of this activation includes longevity, enhanced immunity, and stress resistance. For the last 10 years, we have been publishing papers on various aspects of this insulin-adaptation signaling pathway with respect to aging and longevity. Today, I'll be talking about two recent papers that have been published last year. The topics that are covered by these papers are rejuvenation and healthy aging. The first paper I'm going to present is about rejuvenation in C. elegans. Because C. elegans is one of the most important model organisms for making new discoveries in aging research field, we asked whether we can use C. elegans as a model for organismal rejuvenation. For this, we focused on immune aging, immunosenescence, and reversing this process, immune rejuvenation. Let me briefly introduce the concept of immunosenescence. Immunosenescence is an age-dependent decline in immune functions. Aging increases susceptibility to infection and reduces immune surveillance. Immunosenescence seems to be irreversible in most living organisms, and immunosenescence is a major cause of high mortality in the elderly, and therefore it is very important to understand the immunosenescence at the molecular and organismal levels to combat pathogens in old age, such as COVID-19. Can we study immunosenescence in C. elegans? The answer is yes, and I'll briefly introduce an established experiment paradigm of C. elegans immunosenescence assays. Young or old animals are transferred from normal food, E. coli OP54 C. elegans, to pathogenic bacteria, Pseudomonas aeruginosa P14. And we measure their survival and signaling over time. Previous papers have shown that the mean survival time of animals on PA14 and immune signaling activity gradually decreased during aging. Conversely, PA14, the pathogen accumulation in the intestinal lumen, increases with age. These findings indicate that C. elegans displays immune aging, immunosenescence. However, the relationship between longevity and immunosenescence remained largely unknown, and we wanted to determine their relationship. First, we confirmed that old wild-type worms shown in red displayed shorter survival on PA14 than young wild-type worms did, as shown in black. Very interestingly, the DAF2 mutants that I introduced in the introduction show opposite phenotype. In other words, we found that DAF2 mutants, insulin-adaptation receptor mutants, displayed an age-dependent increase in survival on PA14. By using temporal RNA interference targeting DAF2, we show that genetic inhibition of DAF2 in the middle age of C. elegans was sufficient to further increase the survival of old animals on Pseudomonas. So these data suggest that DAF2 insulin-adaptation signaling receptor mutations reverse immunosenescence. We then asked which downstream factors mediate this reverse immunosenescence caused by reduced insulin-adaptation signaling. This is again the simplified insulin-adaptation signaling pathway that regulates longevity, immunity, and stress resistance. As I told you before, transcription factors DAF16, SKIN1, NRF, HSF1 contribute to these processes. Among these, we found that FOXO, DAF16, and HSF1 were required for the reversed immunosenescence in DAF2 mutant, and I will show you the supporting data in next slides. We found that mutations in FOXO suppress the reversed immunosenescence in DAF2 mutants. In addition, the expression of several DAF16 target genes, including this one, was substantially increased during aging in DAF2 mutants. We obtained similar results for HSF1. So these data suggest that FOXO and HSF1 contribute to immunocompetence in old DAF2 insulin-adaptation receptor mutants. As FOXO and HSF1 are both transcription factors, we then asked whether any of transcriptomic changes responsible for enhanced immunocompetence in aged DAF2 mutants. To address this question, we performed RNA-seq analysis comparing young and old wild-type and DAF2 insulin-adaptation receptor mutants. Because wild-type and DAF2 mutants displayed opposite immunosenescence phenotypes, we focused on genes whose expression was oppositely regulated during aging between wild type and DAF2 insulin-isoform receptor mutants. We functionally characterized these genes by using genetics and found that one such gene, IN7, insulin-like peptide that acts as an agonist of DAF2 insulin-isoform receptor, was an important target. As you can see here, IN7 expression was increased in wild type during aging, but decreased in DAF2 mutant aging. So we confirmed this result by using an IN7 promoter-driven GFP transgenic animal. We then found that FOXO mutations increased the IN7 expression level in wild type and DAF2 mutants, and also found that HSF RNAi did a similar thing. Next, these data suggest that IN7 expression is negatively regulated during aging in DAF2 mutants by FOXO and HSF1. That's the reason why mutations increase the level. Next, we asked whether IN7 contributed to immunosenescence or immunocompetence. Importantly, we found that IN7 mutation increased PA14 resistance even at old age. So overall, these data suggest that age-dependent decrease in IN7 expression in DAF2 mutants underlie delayed immunosenescence in DAF2 mutants. Let me summarize this part with the models. So we found that compared to young DAF2 mutants that displayed enhanced immunity, in old DAF2 mutants, this immune signaling activity is amplified. That is because increased FOXO and HSF1 activities in DAF2 mutants gradually decrease IN7 expression, and subsequently further reducing the activity of DAF2 insulin-isoform receptor. I forgot to mention that DAF2 insulin-isoform receptor mutation that we used in this study is not null, so the activity can be further reduced by reduced IN7 expression or activity. So this leads to further activation of FOXO and HSF1 and further downregulates IN7. So this whole positive feedback loop, signal amplification, seems to contribute to further enhanced immunity in aged DAF2 insulin-isoform receptor mutants. To our knowledge, this is one of the first studies showing that genetic manipulation can reverse an aspect of aging immunosenescence in C. elegans. So far, I've talked about the role of insulin-isoform signaling pathway in immune rejuvenation. In the second part of my talk, I'll briefly talk about healthy aging regulated by insulin-isoform signaling pathway. So how can you achieve healthy longevity? That is our main question for this project. Many previous studies have identified various longevity interventions, but the serious issue in many cases, longevity increases time of frailty in lifespan associated with age-related diseases. So therefore, one of the most important goals in the aging research field is to devise proper intervention strategies for increasing healthy period or health span during human lives. Here we want you to identify long-lived animals without health defects, which are critical for developing strategies for healthy aging by focusing on insulin-isoform signaling using C. elegans genetics. As I told you in the introduction, mutations in DAF2 insulin-isoform receptor promote longevity, enhance immunity, and stress resistance in C. elegans, but generally impair fitness and health, such as motility and reproduction. In addition, DAF2 insulin-isoform receptor mutations cause defects in development called DAWAR, constitutive hibernation like larval arrest at 25 degrees Celsius. In this project, we asked whether we can retain these beneficial traits from a human perspective in DAF2 mutants while suppressing these unfavorable phenotype using the power of C. elegans genetics. Specifically, we asked this question: can we isolate long-lived insulin-isoform receptor mutants with less unfavorable phenotypes? We performed Ems Mutation Screen using DAF2 mutants with 3 million haploid scale. Our strategy is as follows. Among these 3 unfavorable phenotypes, we used Dower phenotype as a representative one for suppressors because it is the easiest. So we screened mutants that did not display a Dower phenotype at 25 degrees Celsius and immediately moved many of these mutant worms and performed survival assay on a pathogen, Pseudomonas aeruginosa, that I introduced in the first part. And we selected worms with enhanced immunity. We use this pathogen survival assay that takes relatively short time as a proxy of lifespan assay because this way we can establish fertile survival lines. We verified one of the fertile mutants survived substantially longer than wild type on the pathogen. So what is this mutation? We found that the isolated mutation is a missense mutation in PTEN, which causes an evolutionarily conserved cysteine 152 tyrosine change. Hereafter, I call this as a PTEN-C2I. In C elegans, the name of the PTEN gene is DAF18. In insulin-like signaling pathway, PTEN, lipid phosphatase, acts antagonistically to PI3 kinase, and PTEN converts PIP3 to PIP2. And this leads to the transactivation of various longevity promoting transcription factors. As we identify this PTEN-C2I from the pathogen resistance assays, we asked, does PTEN-C2I retain long lifespan without impaired health in DAF2 mutants? We first measured the motility span of the animals by using swimming assays. Interestingly, we found that DAF2 mutants with PTEN-C2I, shown in red, increased the motility in young animals compared to DAF2 mutants shown in blue, but retained the motility for a longer time than wild type, shown in black. We also used strong loss of function PTEN mutant as a control, PTEN null for simplicity, shown in purple. We found that PTEN null mutants substantially decreased the motility in insulin-like receptor DAF2 mutants at both young and old stage. So the main point is DAF2 mutants with PTEN-C2I move well throughout lifespan. Let me show you actual movie clips of DAF2 mutants, DAF2 PTEN-C2I, and DAF2 and PTEN null. The movie doesn't work very well. Yeah, here we go. So you can see that at day 3, these DAF2 mutants with PTEN-C2I move much more vigorously than the other two. Okay. So these data suggest that, again, confirm the benefit of a PTEN-C2I. In addition to this movement, we found that DAF2 with DAF2 mutation with PTEN-C2I lived substantially longer than wild type, and PTEN-C2I also restored development, reproduction, and feeding defects in insulin-like receptor mutants. So overall, these data suggest that DAF2 mutants with PTEN-C2I are long-lived and healthy. What I showed you so far is PTEN-C2I retained increased lifespan and conferred normal fitness health in DAF2 mutants, whereas PTEN null mutation decreased both lifespan and fitness. To investigate the underlying mechanism why these two mutations are different, we first asked whether PTEN-C2I affected the enzyme activity of PTEN. We found that PTEN-C2I affected the enzyme lipid phosphatase activity substantially. And we also measured the protein phosphatase activity because previous papers showed that PTEN can act as a protein phosphatase as well. We found that PTEN-C2I retained the partial activity of protein phosphatase. So these data suggest that perhaps remaining protein phosphatase activity of PTEN-C2I the PTEN-C2I may underlie these uncoupled phenotypes, although we have to identify functionally important specific protein substrate in future research. We then asked, does PTEN-C2I and PTEN null differentially affect downstream regulators in insulin signaling? To address this question, we analyzed transcriptomic changes caused by PTEN-C2I and PTEN null in DAF2 insulin receptor mutants by using RNA-seq analysis. The red circles indicate genes whose expression was changed by PTEN-C2I, and the purple ones indicate genes that were changed by PTEN null. Among the comparisons with various published data, we found that three of the factors were differentially affected by PTEN-C2I and PTEN-NUR, shown here. For the sake of time, I will explain the results by using this model. What we found was PTEN-C2I retains the mild activation of FOXO while restricting PMK to NRF signaling axis in DAF2 mutants compared to PTEN-NUR. We confirmed this RNA-seq analysis by using FOXO-GFP and NRF-GFP nuclear localization assays. Next, we asked this question: does the tuning of FOXO and NRF activities by PTEN-C2I have causal effects on healthy longevity in DAF2 mutant by using genetics? So we addressed this question by altering the activities of FOXO and NRF oppositely from this model and see whether that changed the phenotypes. Consistent with this model, we found that genetic inhibition of FOXO robustly decreased the longevity of DAF2 and PTEN-C2I animals. We also showed that gain-of-function mutation of NRF decreased the lifespan of longevity of DAF2 PTEN-C2I double mutants. Conversely, inhibition of PMK or NRF increased the lifespan of DAF2 mutants with the PTEN-NUR and also increased motility in middle-aged animals. So these data indicate that hyperactivation of the signaling axis is the reason why PTEN-NUR mutation decreased lifespan, and suppression of these is contribute to the longevity of DAF2 mutants with PTEN-C2I. So overall, these data suggest that proper calibration of FOXO and NRF by PTEN-C2I contributes to healthy longevity. To sum up, by performing a large-scale unbiased mutagenesis screen, we identified a specific mutation in PTEN that uncouples longevity from health defects in DAF2 mutants. We show that a proper modulation of FOXO and NRF activities calibrated by PTEN-C2I promotes healthy aging in animals. We also found that hyperactivation of NRF, which was supposed to be a good thing in many cases, can cause harmful effects on health span. So our results indicate that by altering the activity of single component in a signaling pathway by a point mutation, we may be able to achieve healthy longevity in animals, and this may be perhaps applied to humans in the future. I'd like to finish my talk by thanking all the people in my lab and our collaborators. And the first part of my talk was mostly done by Eugene Lee and Yoonji Jung, and the second part was done by many people, including Usun Hwang and Haeun Park. And Seok-Jin Ham did almost all the RNA-seq analysis, and Euna Kim did biochemistry essays. Thank you for your attention, and I'll be happy to take questions.
Thanks, Sanjay. I think it's a really interesting talk. It's, trying to wire together all the pathways that are controlling aging and other health span phenotypes is an interesting challenge. And worms are like a great opportunity to do that. I want to start with a very general question, and then we'll get to more details. I feel like that based on how funding is going these days, that it's getting harder and harder to justify getting money for non-vertebrate organisms, yeast, worms, flies, Hydra for that matter. I think that, we both probably believe there's still a lot to be learned using these systems. So maybe you could articulate that and tell us, where you think the advantages of worms specifically are today compared to doing everything in human data sets.
Right. So that's a great question, and I always think about that, question too. And so, for example, my second part of my talk, nobody would imagine that PTEN tumor suppressor mutation can actually cause healthy longevity in a specific condition like DAF2 mutant. So, actually, in our paper, we already discussion points that PTEN-NUR, that point mutation may cause cancer in normal people, but maybe combining with type 1 diabetes, it can have a beneficial effect. nobody would have thought about that. And with the mammalian study, so that those are, those like new findings, unexpected findings are major points of the studying C. elegans, yeast, and Drosophila.
I'm wondering how conserved that is. Is that site in PTEN conserved in mammals, and it'd be interesting to do some mouse studies on it if it is.
Right. So it is conserved, and in humans there is a case that it causes some syndrome that has many problems of cancer. So that is relevant, and I skipped that part because of for the sake of time. But actually, in C. elegans, the single mutation does worse than the wild type. So only combination of DAF2 mutation and the PTEN C2I have a beneficial effect. Yeah.
So that gets back to what I think you were just saying, that there may be that in those individuals that have those mutations, manipulating insulin IGF signaling could be an effective, I don't know if I want to say therapeutic, maybe preventative approach to some of the conditions.
Right. Yeah. Potentially. But one of the reviewers didn't like that, so we took that out. But I really like that discussion, actually, because, you never know, right?
Yeah, you never know. But it sounds like a good experiment to do in a mouse model.
Right.
Because, it's not that difficult to manipulate, insulin IGF signaling pharmacologically or even with lifestyle. So, that could be a really intriguing approach for people with this syndrome.
Yeah, maybe I have to do that because, like you, I started working on mice and mammalian systems as well. And the biggest reason is that my funding agency, again, doesn't like me just working on just C. elegans. C. elegans. So I have to expand my research area to mouse and mammals. And this is really fun, actually, studying.
Yeah, it's a learning process. I'm glad we went into, for those who don't know, we were doing totally yeast and then a little bit of worm work, and then I don't know, a long time ago now we switched over and started doing a lot of mouse studies, and it was definitely a learning curve involved, but it was definitely worth it too, because we've learned a lot from using that system, and I think the ability to go back and forth as we're describing is really important as well. You mentioned immune system in C. elegans, and so maybe for people who are not aficionados, tell us a little bit more about what an immune system means. I mean, C. elegans don't have adaptive immunity,
right? So
you're talking about innate immunity, and how does insulin IGF regulate innate immunity?
Right. So as Brian said, C. elegans doesn't have a specialized immune cells like B cells or T cells, but have innate immune systems. And major immune barrier is first the epidermis, so it just block the one of the major one is block the pathogen, and second one is intestine. So intestine expresses many antimicrobial genes, and one of the main signaling pathway that was established by Dennis Kim and other people is a P38 MAP kinase pathway, and that is important for mammalian innate immunity too. So that has some conservation from the C. elegans to mammals. So those two organs, in my opinion, so epidermis and the intestine, block the combat against pathogens.
It would also seem like you can learn something about sort of the relationship between microbiome and immunity, which is a hot topic in mammalian systems right now from doing this, because there's bacteria, or they're in the gut too. So it's
that's true. So many research groups have been working on the host microbiome interaction using C. elegans and many bacteria. And the great thing about C. elegans system is you can genetically manipulate both host and the pathogen or microbiome relatively easily compared to mammalian system. So I think there is an advantage of that.
I was wondering about the DAF2 effect, that it's protective from the pathogen. And DAF2 mutants are also long-lived. And so when you see something like that, you'd say, are they protected just because they slow aging, or is there a separate phenomenon going on there? In other words, when you take two worms of the same age and one of them is DAF2, it's actually a younger worm. And so I'm wondering if it's a DAF2 effect or if it's just an indirect effect of the worm being younger. Did you try to sort out those two things?
That's a great point. And so we worried about it. And actually, one of the reasons why we started working on that is when we did the pathogen resistance assay, using young worms, we didn't have a big effect with DAF2 mutants compared to the published work. So in published paper, there was a huge increase in the survivor. And when we used the young adults, we have like a 20% increase. And it was consistent. And then we realized that old worms, old DAF2 mutants do better than young DAF2 mutants. So that is the basic.
So the DAF2 mutants are really going in the reverse direction.
Right. Not compared with the wild type, not compared to young DAF2 mutants.
Yeah. That's an unusual phenotype.
Right. So that was the reason why we started working.
Yeah. And I think that gets to one of my next questions was, a lot of the work in worms and for that matter, flies and yeast has been, doing, entire lifespan, some genetic manipulation and showing that the animals live longer. In worms, it could be from the egg or it could be from the L4 stage, but usually it's the entirety of adulthood. And so you were touching on this idea of rejuvenation, and I'm wondering sort of, Micah Petruszek's done work on this too. I'm wondering, what the spectrum of long-lived mutants will also have a rejuvenation effect in worms, because there are lots of long-lived mutations.
Right.
And how common is rejuvenation, do you think?
In C Elegans field, I don't think there are many. So Cynthia Canyon's group showed that aerobic starvation and the resuming the development has some sorts of rejuvenation process. And recently, like we did using DAF2 RNAi from the Middle Age, some people use the AID, the oxygen-inducible degradation system, for knocking down DAF2 in middle-aged adult forms and show some features of rejuvenation. But there are not many studies actually compared to mammalian mouse studies. So C Elegans rejuvenation studies are behind, I guess. So
yeah, I mean, it might make sense that a mouse a mammal has more resilience and ability to rejuvenate with a lot more stem cell populations available to it. So it's possible that the C elegans is going to be much more restrictive in terms of rejuvenating mutations than mammals are, I think. But it's an interesting question. I was also wondering on the N7, the insulin-like peptide. First of all, if memory serves, there are a lot of insulin-like peptides in worms, right?
Right.
So my question was, does N7 have other phenotypes that might give clues to what it's doing related to immunity?
Right. So N7 was first shown by Colin Murphy when she was a postdoc in Cynthia Canyon's lab by using microarray. So she found that N7 level was down in DAF2 mutants, and she showed that N7 RNAi extends lifespan. So that was the first one. And Manhua Tan's group showed that N7 mutation actually increased the pathogen resistance. And I think there are some behavioral phenotype as well. Maybe I'm wrong. So that was already known. What is new about our finding is N7 mutation in old age were still resistant to the pathogen. So that was the new thing. So as you correctly remember, C Elegans has a 40 insulin-like peptides, and N7 is one of the best characterized one out of those 40 in terms of longevity and immunity. So that is that's the reason why we found it, I guess.
Do you think this property is unique to N7, or do other insulin-like peptides share phenotype?
So other insulin-like peptides may have roles. We didn't test in our study. And one of the issues of studying insulin-like peptides is because of redundancy. Sometimes you don't see phenotype by using just a single mutation. So maybe we missed a lot because of that.
You have to knock down all 40.
Right, or study using overexpression.
I had one other question that's more detailed, and then I'll try to bring it back a little bit. You were able to separate the dour phenotype from the immunity phenotype. Can you separate the longevity phenotype from the immunity phenotype? I mean, you didn't screen for that, so I'm just asking you to speculate. So
that's a great point. Not for C elegans, but other people recently showed that some of the longevity mutants have susceptibility to pathogen, and that was shown by Arjuman Ghazi and other people. So I thought those two things are really tightly correlated. Mm-hmm. That's the reason why I did a screen like that. But recently some papers show that it's not always the case. Not C elegans. I mean, not
even the normal bacteria, as long as it's alive, is a little bit pathogenic to the worms in late life, right? Right. Yeah. So I was wondering how correlated those two phenotypes were.
Yeah, they are correlated, as you said, because one of the main reasons why C elegans die when they are old is because of the infection by E. coli. So they are infected by their own foods. So it's like a normal flora attack old age people. It's just a similar concept.
Well, increasingly humans are killed by their own food too. So that's maybe not that, or their choices of food anyway. So,
very similar.
So what's next? Where do you see your research going? What excites you these days?
So these days we are, as I told you in the introduction a little bit, we are more into the non-coding RNAs and RNA quality control. And actually, the major reason is I got funded for those kind of projects. And so we are very interested in novel RNAs like circular RNAs and tRNA-derived small RNAs and double-stranded RNAs and how they work in aging process. Very, very interestingly, we found that many of those strange RNAs accumulate in aged organisms. So last year, we published a review paper showing that not all the time, but in many cases, circular RNAs accumulate in aged organisms. C. elegans, Drosophila, some mammalian brains. And we think that's because circular RNAs are circular, so it cannot be degraded cleaved by exonucleases, so they are more stable. But we still don't know whether accumulation of circular RNA in aged organisms is beneficial or detrimental. So that's what we are really interested in.
Well, it also could be increased synthesis too. I mean, I get the stability argument, but I wonder whether there's increased synthesis too due to
due to the imprecise splicing. Yeah, that's a possibility. Right.
Okay, so I'll quit talking and bring Charmaine in, who I've already introduced earlier, and she's not nearly as scary as I said, so don't worry.
Hi, Prof Lee. Great talk. I have a couple of questions from the audience. So I think what I'll do is I'll ask you more specific questions, and then we'll go towards a broader base question. Okay. So the first question is, do you know or can you speculate whether mutations in the insulin signaling pathway observed in humans or rodent studies can lead to similar benefits on immunosenescence or fertility as you see in the nematodes in your study?
Right. At least in Drosophila and mice, it's been known that similar mutation, mutations in insulin or IGF-1 receptor extends lifespan. So one can do similar experiments. But as far as I know, nobody has published that because we published last year. Maybe somebody is working on that. So it is totally possible. For humans, I don't think it's possible, right? It's unethical. But we can do some case studies because Yushin Seo and other people show that polymorphism in IGF-1 receptor is associated with longevity. So we can see whether they are, we can see their infection records and whether they did better when they're old compared to young. But I think it's going to be very speculative. for human study.
Might be an interesting angle to look at maybe in the future. So the next question is, what are the immune factors in the C elegans that are controlled through FOXO in your study?
I see. So so that's a great question. So we found that INS7 to DAF2 insulin receptor to FOXO, the circle, contributes to immunity. And we believe there is another factor called CYP10, another transcription factor that also regulates immunity. For the sake of time, I just deleted the part because it's too complicated. So we found another transcription factor, CYP10, and that contributes to longevity. But what we couldn't find in our study is there must be some kind of antibacterial peptide or effector peptides that actually work as antibacterial, antipathogen protein, but we couldn't find that in our paper. But previous papers show that some lysozymes or peptide that directly bind to, possibly directly bind to a pathogen may kill or may impair the pathogenesis in C elegans. So those are already known, but what we couldn't find is the specific immune peptides for our immunosenescence assay. Yeah.
Thank you. So I think what I'll do is that I'll move on. There seems to be some people asking very specific questions about PTEN, which you covered in your second part of the presentation. One person asked, is the tumor suppressor activity of PTEN dependent on its lipid phosphatase activity only? And if this is the case, then the mutant affecting protein phosphatase could still retain the TS activity but increase healthy longevity. It's a very specific question.
That's a very interesting angle. So I did, because I'm a C elegans researcher, I did limited literature search for writing the paper. As far as I understood, the majority of the studies are focused on lipid phosphatase activity of PTEN. But some mammalian studies show that protein phosphatase activity also is important for PTEN's tumor suppressor activity. But if I guess, I think lipid phosphatase activity is more important. So the question or comment is, I think I agree with the comment. So it may be really possible to just manipulate protein phosphatase activity without disrupting lipid phosphatase activity. We may be able to suppress the tumor still, but may have a beneficial effects on healthy longevity. And that's a great point.
And a lot of people working on PTEN, it seems, because there's a lot of specific questions. So the next question is, could you expand on the type of tests used for testing PTEN's protein phosphatase activity? And was this using FAK as a target or substrate?
Right. So we use the general peptide for protein phosphatase activity that was already known. I forgot specific peptide, but we wanted to identify a specific protein substrates in C elegans. And FAK, I don't think we test that, but we tested several of them using potential protein substrates genetically, but we didn't see phenotype, so we didn't test that. So short answer is we didn't use FAK, we just used the general genetic peptide for phosphatase activity assay. Yeah.
Okay, thank you for the answer. I think what I'll do is now I'll move towards more general questions away from the more specific ones.
Like a PTEN revolution.
Yes, seems like a lot of PTEN experts in the audience.
Yeah, I think they're more expert than me.
Okay, I think I'll ask a general question then. So as you know, for aging, there's hallmarks of aging, right? So in your with your expertise in C elegans, do you think there's any hallmarks of aging that you can't use C elegans as a model organism for?
We can't, or we can?
Cannot.
Cannot, sorry.
We cannot. Okay, I see. So What are the hormones? I forgot. I have to remember. So some like telomere shortening is not, there is one paper showing that telomere length is correlated with longevity, but otherwise it's not going to be the main issue in C. elegans. What else? So proteostasis is there, and genome integrity is there. We show the inflammation. Inflammation. I don't think anybody defined inflammation in C. elegans well. So those are some parts are not hard to claim to use C. elegans as a model for studying the hallmarks of aging.
And I think in the same breath of questions, in the course of your work, do you encounter any limitations working with C. elegans?
Right. So C. elegans, one of the biggest issue of C. elegans is after becoming adults, after finishing development, somatic cells are post-mitotic. So they don't proliferate. Only the germ cells proliferate. So it's hard to test the proliferative aging. So some people, it's hard to study cellular senescence, for example, in C. elegans because the other cells, somatic cells, don't divide. So there is one big issue, I guess. And I think that criticism that criticism exists in for my postdoctoral advisor, Cynthia Kenyon. So she also told me. So some people think that C. elegans is not a great system for studying aging because of those limitations. But it turns out that many of the genes that regulate C. elegans aging are conserved in humans, right? So that's the argument against that. But still, I think still cellular senescence is difficult to study. And that's one of the reasons why we started working on both C. elegans and senescent cells for studying a new gene. So when we find a new gene that regulates aging in C. elegans, we want to test whether that is important for cellular senescence using mammalian system. Because, for example, we think RNA quality control is important for delaying aging organismally, and also because it's the quality control, like homeostasis, it should be important for preventing premature cellular senescence too. So we test both systems these days, and surprisingly, in many cases, the genetic manipulation that increase or decrease C. elegans lifespan have similar effects on cellular senescence too. So I think by using those systems, we can overcome those limitations. Yeah, that's one.
This is Brian. I still think it's surprising that how conserved their aging is among these different organisms. I mean, this is something that when we started in yeast, I didn't really suspect what we would learn in yeast to be conserved, even in worms, or much less in humans. And yet we keep stumbling into these levels of conservation. So do you have a thought as to what that means? I mean, why do worms and yeast and flies and humans all seem to age in at least overlapping ways?
Right. So, I mean, that's still a mystery, right? But I guess the aging is ancient. And as the previous speaker, I forgot her name, I'm sorry. Hydra has some similarity with the other system, right? That was really interesting and surprising. So I think aging is a really ancient and fundamental biological process. So no matter what the details are, so there are fundamental similarities. And that's the reason why we still think C. elegans is a great system. And in fact, many possible longevity promoting drugs like rapamycin and metformin have been shown to work in those simple model systems, right? And it turns out to work in mammalian system and respiratory too, right? So all those.
I was with you for a long way there, Sanjay.
Right, definitely. So yeah.
I agree though, generally. Charmaine, one last question. Make it good.
Oh, shall I just ask more fun question.
Yeah.
So seems like a recurring question on every episode, but people are very interested to know, Prof Lee, do you personally have any regimes or supplements or anything you do for healthy longevity yourself?
Yeah, you haven't aged a day since I met you, so you must be doing something right, Sanjay.
No, I dyed my hair. So cosmetic surgery. I'm kidding. So I don't personally take anything, but I think I do a little bit of intermittent fasting. So I usually don't eat breakfast, and I sometimes skip lunch. So it's one meal. I try to eat just one meal a day. So that's what I do, because that's the easiest. Other things are difficult, like dietary restriction. Constant dietary restriction is impossible for me.
Well, you're going to come in October to give a talk in Singapore, and so we'll try to make that one meal a really good one when you're here. So thanks a lot for the talk. Thanks, Charmaine. And I want to remind the listeners to use the chat function and the panelists and all attendees option to leave comments and feedback. Check out the end credits, because it may have news from our center or the School of Medicine. We now have a Twitter account, and there's a QR code, and you can find it at the end of the webinar. And we recently launched our Center for Healthy Longevity website, and there's a QR code for that too that will be available shortly. We're taking a week off next week. We'll have Professor Eva Hoffman on August 4th with Andrea Meyer. And she'll be talking about a topic which overlaps between our Center for Healthy Longevity and our Asian Center for Reproductive Longevity and Equality, and that's reproductive longevity and ovarian aging in women. With that, I want to leave you with some thoughts from Neil deGrasse Tyson on the possibility of living forever. Thanks again for joining us.
If you could live forever, would you? Sure, that's an attractive idea. But the way I look at it is the knowledge that I'm going to die that creates the focus that I bring to being alive. The urgency of accomplishment, the need to express love now, not later. If we live forever, why ever even get out of bed in the morning? Because you always have tomorrow. That's not the kind of life I want to lead.
Don't you fear not being around?
I fear living a life where I could have accomplished something and didn't. That's what I fear. I don't fear death. You know what I want on my tombstone? Be ashamed to die until you have scored some victory for humanity.
Like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.